Metabolism can be one of the more difficult topics when it comes to biology and nutrition. So I wanted to make this short little video to kind of give an overview of metabolism to supplement or to help kind of condense some of the information that's in the book. So we ended the last video about digestion and the proteins and lipids with the monomers arriving in the cell. So basically, we have glucose arriving in the cell from the blood. So these are coming from the blood and going into the cell through those glucose transporters that were stimulated by insulin. So we have glucose coming from the blood and going into the cells. Almost all of our cells are going to be using glucose, and there's a few cells that can only use glucose, like our red blood cells, and a lot of our nerve cells can really mostly use glucose for energy. And then, so we also had, what do we also have? We had also had amino acids that were traveling from the blood into the cells. And then remember, we had those lipoproteins that were delivering lipids into the cells as well. So I talked a little bit about this last time. These could be used to make glycogen. If there's excess glucose that needs to be stored, glycogen would only be found in the muscle cells and the liver. Or it can be used for energy, and that's the process we're going to talk about today. Amino acids, the same thing, could be used to make new proteins, or could be used as a source of energy if there's plenty of protein available. Lipids, same thing, could be stored as fat or in those adipose cells especially, but also in muscle cells, could be stored as fat. And also, if energy is needed, can be used for energy. So we're going to focus on, last week we focused on that first process. This time we're going to focus on the process of converting the energy that's stored in those molecules into energy that can be used by the cell, mostly in the form of ATP. So we're going to start with looking at the structures. I'm going to focus mostly glucose and lipids as a source of energy. Proteins are more of a secondary source of energy. The cells will preferentially be using glucose and lipids as a source of energy. One of the reasons why is if we look at the structure of glucose and the structure of a fatty acid. So this is glucose here. This is a fatty acid here. We'll see that on these chemical structures, every unlabeled corner represents a carbon. So these are all carbons. There's 6 carbons in glucose. So we have the formula C6H12O6. And then it's implied that there's also the correct number of hydrogens attached to make a total of 4 bonds for each carbon. So I think we need one more here. So that's our glucose molecule. The fatty acid, the same thing. So these are all carbons here. And then they would be attached to the appropriate number of hydrogens. I think I went through that in the last video as well. So what I want you to notice, though, is that in the glucose molecule, let's focus just on glucose for the moment. We do see a lot of CC and CH bonds. And this should be something that's familiar to you from biology or chemistry. These are nonpolar bonds. Nonpolar bonds are less stable, and that means they have more energy. The analogy I like to give is if you had a seesaw and it was perfectly balanced, it's more likely that it's going to move right back and forth. That's the equivalent to a nonpolar bond, because remember, covalent bonds are the sharing of electrons, and these are sharing electrons equally. So the electrons could be found equally likely around either atom. And that's in comparison to a polar bond. So this is nonpolar. if you had a seesaw and you had something or buddy that was much heavier than the other thing or the other person on the other end, whoops, what you would find is that this is just going to be sitting on the ground, right? And this side is just going to be up. It's not likely that it's going to be wobbly. So this is representing or kind of an analogy for a polar bond in which the electrons are shared unequally. An example of that would be a carbon-oxygen bond, where the oxygen's more electronegative. So the oxygen is going to the electrons are going to be almost always going to be found nearer to the oxygen atom than the carbon atom. So that's going to be much more stable, and therefore there's less energy stored in this bond between carbon and oxygen. Okay, hopefully that's a little bit of review for you, but a lot of people need that review too. So if we look at the reactions where we are releasing energy, what we're going to be doing is we're going to be replacing nonpolar bonds with polar bonds in reactions that release energy. And this process is called oxidation. Oxidation literally means like the loss of electrons. So when you go from a nonpolar bond like CC or CH, and you go to a polar bond, you can see that the electrons are like moving away from carbon and closer to oxygen. So we can say that the carbon is being oxidized because the electrons are moving away from the carbon. So oxidation is the process that happens during cellular respiration and other processes like for fatty acids, beta oxidation. So the end product of cellular respiration and beta oxidation, which feeds into cellular respiration, is carbon dioxide. And carbon dioxide is like this. So it's the double bonds, 2 oxygens, 1 carbon. And because the oxygen is highly electronegative, the electrons that are shared very unequally, and this carbon is completely oxidized. So you can't really oxidize this carbon any more than that. That's a completely oxidized carbon. So we have taken those nonpolar bonds that stored energy, and now we have this molecule, which doesn't really store energy at all. So what happens to those energy? What happens to that energy? So that's the part that I wanted to focus on in this lecture, is the process of cellular respiration and how those bonds can be converted and how the electrons can be used to generate ATP, which is a form of cellular energy. So one thing I want you to think about is there are electron carriers. So there are many, many reactions, and I'm not going to go through all the reactions. You can look in your book for the reactions, and there's a little video that's associated with the different reactions that are involved in cellular respiration. But I do want to kind of go over this idea of electron carriers. This is especially important nutritionally speaking, and I will be drawing my somewhat funny looking truck here. So I like to think of an electron carrier like a dump truck. And actually a dump truck would have like a platform, and then it would have like a thing on top. And the dump truck can carry 2 electrons. So the dump truck is going to carry 2 electrons away from the carbons as they are being oxidized. So this is this would be an electron carrier. So as carbon is oxidized, electrons are carried away by these electron carriers. And there's 2 of them. There's NADH and FADH2. And you'll learn more about these later in a nutritional point of view, because these are actually derived from vitamins niacin and riboflavin. So the N is actually related to niacin, and the F is related to the flavin and riboflavin, which are both B vitamins. So B vitamins are really important for metabolism. But we're getting a little ahead of ourselves because we're going to do micronutrients later. So basically they're carrying, as carbon is oxidized, the electrons are loaded into the dump truck, and then the dump truck drives away, and it's going to drive to the electron transport chain. In the electron transport chain, you can see here is the NADH. That is that full dump truck. So we've got dump truck with its electrons, and it's going to drop off those electrons to the molecules in the electron transport chain, which are found on the inner membrane of the mitochondria. And as it carries those away, or as it drops those off, it's going to become empty again. And as it's empty, let's see if I can draw that truck again. It's going to drive off. Truck drives off, and it goes back to the reactions of glycolysis, which is in the cytoplasm, and the Krebs cycle or citric acid cycle. So it's going to go back, and it's going to go, and it can pick up more electrons, and it can bring them to the electron transport chain. The function of the electron transport chain is to release the energy of those proton gradient or an H plus gradient that can be used to ATP. So the proton gradient is formed in this intermembrane space. It's kind of like the electrons are passed from molecule to molecule in the electron transport chain. I like to think of it as like a hot potato. So I can actually take hot potatoes out of the oven with my bare hands. I don't know if that's a good thing, but I can. But I can't hold on to them very long because they're too hot. So that's like these electrons that are being dropped off. They're hot. So they are full of energy. So they need to be passed on to the next molecule, just like I would pass the hot potato on to the next person. And as I pass it along, it's losing energy. And as the electrons are passed along, they're losing energy. But that energy can be captured to move protons, move H plus across the membrane into this space. So now you have more on one side of the membrane than you do on the other side, and that's called a gradient. That's the proton gradient. And this itself is a form of potential energy. Those protons are allowed to go back down through this molecule, this enzyme called ATP synthase. And as they go down through ATP synthase, that energy is going to be converted into putting the phosphate group onto ADP. So ATP synthase is an enzyme, and it's catalyzing this reaction of going from ADP plus phosphate to ATP. And this is our form of cellular energy that the cell uses. As the cell uses this energy, that phosphate group will be broken off, that releases energy, and then you go back to ADP plus phosphate, which can be recycled and reused. So this cycle is happening like every minute. Your ATP is being recycled into ADP and remade by the process of cellular respiration. So there's some good, like I said, there's some good animations and videos in your books, and I'll direct you to those in the announcement. But in the meantime, I wanted to go over sort of an overview of all the processes, and I'll provide this overview that I made. It's just a concept map I made of the different processes that are involved in the different types of molecules that feed into the release of energy for our cells for making ATP. So glucose is the easiest preferred form of energy. Glucose is broken down by glycolysis. Glycolysis itself produces some ATP. It's broken down into 2-3 carbon molecules. So really, you're just releasing, you're really producing NADH here. So you're stripping some of the energy, some of the electrons away, but you end up with these 2-3 carbon molecules of pyruvate. So you haven't lost any carbon yet. Then there's a step called the linking step. And in the linking step, one carbon is removed because it's been completely oxidized. So it's removed because there's no more carbon, no more energy left in those nonpolar bonds. And that produces this molecule called acetyl-CoA. And acetyl-CoA is an acetyl group, which has 2 carbons that's attached to a coenzyme A, which you'll learn more about, which is derived from pantothenic acid, which is another B vitamin. And you'll learn more about that later when we do vitamins. So then the acetyl-CoA the CoA is going to carry those 2 carbons into the citric acid cycle, or sometimes called tricarboxylic acid cycle, sometimes called the Krebs cycle. And as it goes through the Krebs cycle, all the remaining nonpolar bonds are replaced with polar bonds, and the remaining carbon dioxide is released. So carbon's completely oxidized, and this produces a lot of electron carriers, NADH and FADH2. They carry those electrons to the electron transport chain, which is over here. So they drop off those electrons like I just showed you. And then the electrons go through the electron transport chain. We generate that gradient. So we've got that gradient, and that gradient can be used to make ATP from ADP plus phosphate. So most ATP is produced there in the mitochondria. One important feature, though, is this process requires oxygen, because that hot potato, or those electrons, they have to have somewhere to land. They can't be hopping around forever. And the place that's a molecule that's very electronegative is oxygen. And so oxygen actually attracts those electrons. And in the process of taking on those electrons plus some protons, it becomes water. Okay, so that's why oxygen is needed. In the absence of oxygen, you cannot have this if there's no oxygen. So if there's no oxygen, you can't make a lot of ATP. Instead, you can only make ATP through the process of glycolysis, which makes just a little bit of ATP. So you can't sustain exercise very long when you need a lot of energy. You can't sustain it very long without oxygen, which is why you get winded, because you're running out of oxygen. Your breathing and your blood and your circulation can't supply oxygen fast enough. So you can only maintain that sort of anaerobic exercise for a short amount of time. And that produces NADH. And eventually you're going to need, because the NADH can't go to the electron transport chain, the NADH will build up. So you need a place for NADH to drop off its electrons. So in fermentation, NADH will drop off its electrons to pyruvate in the process of fermentation. And pyruvate becomes lactic acid. So that is a way for, and as it becomes lactic acid, the NADH becomes NAD+. And NAD+ can then continue glycolysis. So you have to have a place for NADH to drop off the electrons. Okay, so that's glucose. I also have put on this diagram, I've also put how glucose can be made. So glucose can be made from pyruvate. So that's why the arrows are also going in the other direction by the process of gluconeogenesis. I'm going to go over to fatty acids. So fatty acids, because they have so many nonpolar bonds, they have lots and lots of these CC and CH bonds, they can actually produce more energy because there's more nonpolar bonds to convert or to oxidize the carbon from. So carbon's oxidized, and it produces, the beta oxidation process produces a lot of NADH and a lot of FADH2, as each time as a couple of carbons, 2 carbons, are converted into that acetyl group. So it's kind of broken down 2 carbons at a time into this acetyl group, which can feed into the citric acid cycle, and then can produce more NADH and FADH2 that can carry the electrons and make ATP. So alcohol can also feed in as an acetyl group as well, or be converted into an acetyl group. And then the process of making fatty acids is fatty acids are made from acetyl-CoA. This is an important thing to note, because you note that glucose and amino acids have to go through acetyl-CoA. So if there's excess glucose or excess amino acids, they will be converted into that acetyl group, and that can be converted back into fatty acids. So excess glucose and excess amino acids or protein can all be converted into fat. But the reverse is not necessarily true. So you cannot convert an acetyl group. So you cannot convert fatty acids into glucose, because you'll notice that glucose is made from pyruvate, not from acetyl-CoA. There aren't enough carbons here to make glucose from. So That's how those two macromolecules where we get most of our energy are metabolized. I just want to note that we call those processes of making smaller and releasing energy catabolism, and the processes of building up or that use energy and store energy are called anabolism. And then I didn't go into the proteins too much, but amino acids can be converted back to make amino acids. You need to use that process of transamination. So you have to have some amino acid to transfer an amino group to make a new amino acid. But amino acids are used for energy. They have to be deaminated, which means that their amino group is removed. So that nitrogen group is not used for energy. It's only the carbon skeleton that's used for energy. Okay, and then once it's deaminated, depending on the amino acid, it could become pyruvate or it could become the acetyl group. But one thing I want to point out here is that, oh, and then I said excess glucose and excess amino acids can be converted into fat. That's also true for alcohol because everything goes through acetyl-CoA. But you'll notice that the only thing that kind of goes back up into a macromolecule is to fat. So every excess is going to go into fat, is going to be converted into fat. All right, so I hope that helps. I'll provide this document and it might come in handy later when we do the B vitamins and you can map the B vitamins onto this document.